Good morning, everyone. So today we will see the second topic in probiotics and functional foods course. Myself, I'm Dr. Indira. So second topic is the safety of probiotic microorganisms. So why we need to consider the safety of the probiotic microorganisms means, so already we know that probiotic microorganisms are the beneficial microorganisms when administered into the host, they confer the health benefits. So when we are taking those probiotic microorganisms, safety is matter. Safety is important. So in the last 20 years, the number and quality of clinical trials for assessing the probiotics and their health benefits have grown substantially. And the efficacy measurement have been integral to the growth of the category. So here, if you see the safety of probiotic microorganisms have been raised by clinicians and then researchers and then policymakers. Why? Because if probiotic microorganism, if we consume into the body, if it creates any adverse effects, so it leads to the various side effects in the organism, the host, particularly the human beings who are consuming. So here we need to consider whatever the safety aspects of the probiotic microorganisms. So particularly the probiotic strain you have to see, and whatever the probiotic product you are preparing, you have to see the probiotic product, the quality without any contamination, and how you are administering this probiotic product into your body. That administration is also very, very important in terms of safety. So a foundation to assessing the safety of any given probiotic strain is a complete genome sequence. That means you have to study the complete genome sequence of the probiotic microorganism. So here, this allows the assignment of the strain to a given taxonomic group, enabling a review of published risks associated with the species. So whatever the organisms which we are calling it as probiotics. So the scientists, they study about this particular organism and they'll generate the data so related to the risks associated with that particular species. So you have to go through while selecting the probiotic organism. And FL genome sequence, so it allows strain level identification and it provides the entire information of the particular strain. So what are the any toxin genes are present, any virulent nature of the or strain will be present, it is producing any beneficial metabolites or not. So entire data will be available in the whole genome sequence of a particular strain. So it gives the genes of concerns, toxicity, pathogenicity, and an antibiotic resistance. So now scientists are still evaluating the real transfer risk, the clinical and public health implications of such a transfer. Some phenotypic testing is also a component of assessing safety of the probiotic strain. So these are the various aspects we need to study about the probiotic strain and its safety. So if you see the traditional species with the history of safe use, they are strain at the time, strain specific characterization they used to do, and they use those strains for probiotic products. But nowadays, because of advancement of the tools and because of the development of the so many techniques, nowadays we are doing the whole genome sequencing, so we can identify the strain taxonomy and then strain level identity, and then whether it contains any toxin or virulence genes, whether they are present or absent. So all these things we can study because of the whole genome sequencing. Not only this one, when you are considering the safety aspects of probiotic organisms, this particular antibiotic resistance assessment is very, very important. So generally at phenotype level, whether the organism is producing any antimicrobial compounds or not, So whatever the MIC is there, that is the minimum inhibitory concentration within the range for that particular species or not. Generally, you will evaluate at phenotype level. But whereas at genotype level, if your antibiotic resistance genes are present, so you have to see, does the gene encode any resistance to a critically important antibiotic? You have to study that one. Is gene is functional or it is non-functional? If it is non-functional, there is no problem. If it is functional and expressing a particular gene, so then we have to consider its safety. Is gene likely to be transferable? That is transfers from one organism to another organism. So that also you need to consider while identifying or assessing the antibiotic resistance of a particular probiotic organism, and monitoring to the extent possible. That is the adverse effects of these probiotic organisms at acute level as well as the chronic level. Chronic level means long-term effect. So acute level, after taking these probiotic organisms, is there any invasive infection will be there inside the body, or any transfer of antibiotic resistance genes is there? And again, drug metabolism, how it is metabolized in the drug molecules. Is there any microbiome alteration is there? That we have to study. At long-term level, whether this particular probiotic organism inside the gut, whether it is colonizing and attached to the cells or not, that you have to study. And then any microbiome alteration is there, it is replacing any bad bacteria or it is replacing any good bacteria. So you have to study the alteration of this microbiome inside the gut system. And then drug metabolism, how it is metabolized in the drug molecules. Is there any physiological impact on the host? So these aspects you need to cover in long term. Whereas in acute level, these are the various activities like invasive infection, transfer of AR genes, drug metabolism, and microbiome alteration. So these you have to study at the strain level. So after strain level, after studying and assessing the particular strain, whether it is beneficial or not, if it is beneficial, then you can use that particular strain for making the products. So at product level, you have to see the quality as well as the safety of the probiotic product. So whatever the probiotic products you are manufacturing or producing, so those products, whether they are GMP compliant production is there or not, you have to check. GMP means good manufacturing practices. And then safe and efficacious dose. How much dose we have to provide to the host, and whether the product, whatever is manufactured, whether it is safe to consume or not. And again, microbiological testing fit for end user, whether it is fit to consume by the host system or not. So is there any contamination or is there any adverse effect of that particular product? And again, the potency guaranteed through end of shelf life. So what is the shelf life of that particular product? So these aspects you need to consider at manufacturing level or production level. So the main ultimate goal here is the probiotic product, its quality and safety, whether it is used by the consumer or not. And come to the probiotic product administration. So when we are administering, so which route of administration you have to manufacture this particular product. So while deciding the route of administration, and is there any risk with method of administration? So is there any contamination of hospital environment where the products are preserved and stored for consumers, and then contamination of catheters and exposure of nearby patients, and then how dispensed to the patient. So whether it is given by oral or IV or nasojejunal feeding. So the route of administration is very, very important in terms of the safety. And which target population you have to use this particular probiotic and probiotic products. So vulnerable like preterm infants, and then short bowel syndrome, and then obese and pregnant. So we can target this particular products to these vulnerable people. Why? Because of lack of beneficial bacteria in their gut system, generally they are prone to various diseases. So in order to avoid this one, we need to target this vulnerable people by providing this probiotic and probiotic products. The healthy people. So healthy people again, male, female, and what is the age of the people. So all these aspects we need to cover in terms of the safety. And if you see, the most important aspect is the virulent genes, and any harmful metabolites are there. So here you have to see the virulent genes, whether they are present or absent. So generally, if they are present, they encode for toxin production, and they attaches to the host system, and then immune evasion will be taken place. So you have to see whether the strain doesn't contain any virulent genes, that is absence of virulent genes. And again, so this whole genome sequencing need to be employed to detect whether it is producing uh, any metabolites or not. And again, you can observe whether the presence or absence of the virulent genes by studying the whole genome sequence of the strain. So it should not produce the harmful metabolites like biogenic amines. So these biogenic amines, they trigger the adverse and physiological effects like migraines and then hypertension. And the strain should be evaluated for metabolic byproducts that may interfere with the gut homeostasis or triggers the inflammatory response. This is also important criteria. You need to check whatever the metabolites are produced by the probiotics. They should not produce any imbalance in the gut system, or they should not produce any inflammatory response in the host system. And the techniques like HPLC and then gas chromatography are generally used to identify and quantify these metabolites. During production, contaminants and then changes in the metabolic pathways may lead to harmful compound generation, necessitating the stringent quality control measures in the manufacturing units. And these probiotics are further tested under simulated gastrointestinal conditions to ensure that there is no harmful byproducts are produced during the digestion. So the absence of pro-inflammatory substances or compounds toxic to the microbiota is very, very crucial. So you need to perform the simulated gastrointestinal model for assessing the probiotic safety. And then advanced screening technologies like metabolomics, so you can study for identifying what type of metabolites are produced by the particular strain and then aid in comprehensive profiling. So additionally, long-term studies in animal models, it also helps confirms the metabolic safety under various physiological conditions. So this ensures the strain remains safe, not only in controlled environments, but also during prolonged human consumption. And next one, it should be non-pathogen and it is identified as a GRAS. That means generally recognized as safe. So a fundamental criteria for probiotic safety is non-pathogens. So that strain should not cause any infection or any adverse effect, even in the healthy people as well as the vulnerable people like infants and then elderly and then compromised individuals. So to confirm this, in vivo studies in animal models and controlled human clinical trials are need to be conducted so that we can identify whether the strain is GRAS status or not. The strain should not exhibit traits such as invasiveness, hemolysis, or cytotoxicity. If it is there, the blood will be lysed and it is toxic to the cells. So the strain should not carry these activities. GRAS is generally recognized as safe status is a regulatory designation for substances with a proven history of safe use or robust scientific evidence of in their safety, which was this status will be given by the FAO and WHO. Achieving GRAS status involves a thorough review of toxicological data, including acute, subchronic, and chronic toxicity studies. We need to perform and we need to submit this data to the organization so that they provide the GRAS status to the particular probiotic strain. And the strains must also demonstrate the compatibility with the host microbiota, avoiding the other disorders like dysbiosis and unintended interactions. So this GRAS evaluation includes allergenicity assessment to ensure the strain does not exhibit any hypersensitivity reactions. And then safety parameters are evaluated across diverse populations to account for variability in response. That means different age group people and then both male, female are involved in the clinical trials to study the safety of the probiotic organism. And then regulatory agencies like FDA and EFSA require documentation detailing the strain's safety profile and history of use. And adherence to these guidelines ensures the consumer confidence and then simplifies the market approval processes. So it facilitates the safe commercialization of the probiotic products globally in various markets. And antibiotic resistance and horizontal gene transfer concerns. So these concerns are very, very important. Why? Because these probiotics are intended for human consumption. They should not contribute to the spread of this antibiotic resistance genes from one organism to another organism. So this is a very important and critical global health challenge. So to address this ones, the strains are evaluated for intrinsic resistance, which is non-transferable in nature, and acquired resistance, which may pose a significant risk. Genome sequencing is also employed to identify these resistance genes, and we can evaluate the potential for horizontal gene transfer. It is tested using conjugation and transformation experiments under laboratory conditions. And then minimum inhibitory concentration is tested against the standardized antibiotic and different antibiotics. So it identifies is there any resistance profile is there or not. That also we can study using this MIC. And regulatory framework requires detailed reports on antibiotic resistant profiles, ensuring transparency and safety in the probiotic applications. Proper strain selection minimizes the public health risk and aligns with the global efforts to combat the antimicrobial resistance. If you see the regulatory frameworks, mainly they will see the aspects of safety. The safety assessment of probiotics, it is governed by the regulatory bodies like EFSA, FDA, WHO, and other national bodies. EFSA employs the qualified presumption of safety approach, which pre-approves well-documented microbial strains with established regulatory costs. The FDA requires probiotics to attain GRAS status or food additive approval through rigorous evaluation. WHO guidelines emphasize strain-specific assessments, including genetic, toxicological, and clinical data, to ensure probiotics they meet the safety and efficacy criteria. Regulatory agencies mandate thorough documentation covering the strain's origin, genetic stability, and production processes. These are the various activities of the regulating bodies. They identify these parameters and they will assess the probiotic safety of the strain, and finally they provide the approval. In India, we are having the regulatory authority, that is the Food Safety Standards Authority of India. So look after the safety aspects of the probiotic organisms used in the Indian food products. So country-specific regulations are there. Healthcare Canada is there, and India sets a complement education guidelines addresses these safety standards. Compliance with these frameworks not only ensures consumer safety, but also fosters the trust and acceptance in the global market. So advancements in regulatory science have introduced new tools such as unique technologies to refine the safety assessment. So by adhering to these guidelines, manufacturers can navigate the regulatory landscapes effectively while ensuring the integrity of the probiotic products. Thank you.